Good afternoon, everyone. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is a part of our Biochemistry Focus webinar series. Topics in this series include different research areas in molecular biosciences, as well as practical sessions to support career development. Each webinar will focus we will give you an opportunity to ask questions via text, and we welcome suggestions for future topics and speakers for to feature in our webinar series. Please see the website for more details. I would like to take a moment now to introduce myself. My name is Katya Lamber. I did my PhD years ago in EMBL in Germany, European Molecular Biology Laboratory. Right now, I am a senior lecturer at Kingston University, and I am a member of Biochemical Society research area for cells. So that was about myself. Now let's talk about the webinar. Today's webinar is titled New Insights into ER Chaperone Function and Ribosome Quality Control Pathways in Cancer Therapy, and is part of our dedicated early career research program of webinars. We are delighted to provide this opportunity today for John and Susanta to share their work with the molecular biosciences community. Before I hand over to our first speaker, I would like to mention that questions will be asked at the end of the webinar, but please do send in your questions during the talks. If you have a question, please type in the question in the box as shown is an image in the screen, stating who your question is for. And we will try to answer as many questions as possible at the end. Our first speaker today is John Young from the University of Oxford. John is a postdoctoral researcher using native mass spectrometry and other structural methods to understanding how binding of small molecule ligands affect protein function. Before moving to Oxford, John completed his PhD in protein biochemistry at the University of British Columbia in Canada. Today, he will be presenting his work on interactions of the ER chaperone BIP with small molecules and nucleotides, insights from native mass spectrometry. Now I will be handing over to John.
Thanks, Katya, for the introduction. Can you hear me and see the slide?
Yes, I confirm that I can hear you and I can see the slides and I see your laser pointer.
Great. Is the transition working?
Yeah.
Okay, awesome. Well, thanks a lot for the introduction, and thank you also to the organizers for inviting me to present today. I'm going to be talking a little bit about some of the work I've been doing at the University of Oxford recently, using native mass spectrometry to study the function and regulation of the ER chaperone BIP. So what is native mass spectrometry? Native mass spectrometry, actually, before I start, I want to acknowledge a number of people. First and foremost, thanks to my supervisor at Oxford University, Professor Dame Carol Robinson. I acknowledge Drs. Dee Wu, Tarek, Corinne, and Abraham in Carol's lab for a lot of helpful discussions and critical conversations. I also want to acknowledge Professor Christian Wilson at the University of Chile and his PhD student Hilda, who have been really outstanding collaborators on this project. The Robinson Group at Oxford University is based in the Kavli Institute for Nanoscience Discovery, and my research there is supported by a postdoctoral fellowship from the Canadian Institutes of Health Research. So what is native mass spectrometry? Native mass spectrometry is a technique that was pioneered a number of years ago by Carroll and others, wherein globular well-folded proteins in solution, they're exchanged out of tris salt butter into a more volatile butter called ammonium acetate. That protein mixture is then loaded into an electrospray capillary, and under very gentle ionization conditions, the protein is injected into the mass spectrometer in its folded native state. That's really critical because that allows you to observe interactions between protein-protein interactions can be seen in the mass spectrometer, as well as protein-ligand interactions. And that ligand can either be a small molecule nucleotide, it can be a cofactor, or it can be a drug. So native mass spectrometry has a lot of implications these days in drug discovery. What does this look like? Typically, when you perform this workflow, the protein is injected into the mass spectrometer with or without a ligand. So in the case of the APO protein alone, you see this nice, well-distributed series of charge states in purple. If there's a ligand added that binds specifically to the protein, you observe an additional charge series, this add-up charge series shown here, that represents binding of the ligand to the protein. Using publicly available software, that this mass spectrum here can be deconvoluted into this, which shows mass of the APO form and mass of the ligand-bound form. And that can then be, if you then do a titration across varying concentrations of ligand, you can calculate fraction bound from this data and plot a binding curve. Simple example of that is shown here, where I have a purified nucleotide binding domain of a generic ATP binding protein. The protein alone sprays very nicely and produces this charge series here. The mass of that corresponds exactly to the mass of the protein I purified. Addition of ATP across multiple different concentrations, as ATP is added, you see progressive disappearance of the APO form, and with a concomitant increase in the bound form shown in red. And again, the difference in mass from black to red corresponds exactly to the mass of one ATP molecule. So after doing a titration across several different concentrations of ATP, I can plot this binding curve, a fraction bound as a function of total ligand concentration, would allow me to calculate an apparent KD of approximately 3 micromolar for this interaction. So this example here that I'm showing highlights the use of mass spectrometry for studying nuke protein to small molecule interactions. Another thing that you can use this technique for very nicely is looking at protein-protein interactions. So here in this case, I have a purified tetrameric protein called SECB. It's a bacterial chaperone that's involved in the transport of nascent polypeptides from the inside of the bacteria to the outside of the bacteria. So the protein SECB, it is a homotetramer. When you purify the protein from bacteria, you see almost exclusively tetramer at a high concentration of 60 micromolar. But as you dilute the protein down from 60 micromolar to 60 nanomolar, you see disappearance of the tetramer charge series and a concomitant increase in a dimeric charge series. And from that, you can calculate the dimer to tetramer equilibrium of the protein, effectively titrating the protein against itself, and you arrive at a KD here of approximately 170 nanomolar, which is in good agreement with other more classical techniques such as ITC. Actually, yeah. And analytical ultracentrifugation. So that's a little introduction to native mass spectrometry. Why use this method to study BIP? BIP is known colloquially as the master regulator of the endoplasmic reticulum because it's involved in so many essential processes: protein homeostasis, protein folding, protein translocation, calcium homeostasis in the ER, and it's also a promising therapeutic target. Overexpression and misregulation of BIP expression is implicated in development of numerous types of cancers. So in a recent review, there was this very nice model of BIP function. BIP is a protein with two distinct domains: a substrate binding domain that interacts with unfolded polypeptides, and a nucleotide binding domain, shown here in blue, that interacts with ATP. Through a there is a series of substrate and nucleotide dependent conformational changes by which BIP exerts its biological activity, and this is modulated by other co-chaperones called HSP40s, and as well as by nucleotide exchange factors. So I set out to study BIP using native mass spectrometry to see what new insights can I gain using this technique into nucleotide binding and the way nucleotide binding regulates this protein. So that's what BIP looks like again. This is the AlphaFold 3 structure of BIP from yeast. So there's the nucleotide binding domain here shown on the left, and the more elongated substrate binding domain shown here on the right. When I purify this expressed yeast BIP and it's recombinantly in bacteria and purify the protein, there was an unexpected observation almost immediately. BIP purified from E. coli, it exists mainly as a monomeric species, but also a small subset of this exists as a dimer population shown here on the right. But what immediately jumped out at me about this native mass spectrum is, yes, there's the black charge series centered around the 17 plus charge state. That corresponds to the monomeric mass of the BIP protein. But there's also this approximately 368 Dalton adduct, this co-purifying species that comes along with BIP. What could that be? Is it ATP? Certainly not. It's too small. Some studies have found that BIP can be subject to post-translational modifications, so a molecule of AMP can be covalently attached to BIP in a process called AMP elation. Is it possible that that's what this is? No, because the mass is totally wrong. So what is this mysterious co-purifying adduct? This stumped me for a while. And what I did, I actually figured out what this is largely by accident, is I simply changed the expression system. Instead of using this strain, E. coli RR1, I used E. coli BL21. When I produced the protein in that strain, that small molecule co-purifying adduct is completely absent. That led me to suspect that the small molecule co-purifying in the first case was largely because of the strain. Now, the strain of this bacterial strain RR1 has been characterized in some detail. There's a recent study that mapped the genome of this bacteria, and it turns out that this strain harbors a deletion in the metabolism of the nucleotide guanosine monophosphate, the nucleotide GMP. This monophosphate, there's a key deletion in the metabolic pathway of this nucleotide. As a result of that, in this strain RR1, there's an elevated level of GMP floating around in the cytosol, which is why it's co-purifies in this case, but not with BL21. To validate that possibility, I purchased exogenous guanosine monophosphate and added it to the protein. Gratifyingly, between BIP APO and BIP incubated with guanosine monophosphate, there is significant binding taking place, and this mass, this charge series here that I observed, overlaps exactly with what I saw before, which is strong evidence to suggest that this is that ligand. In order to see what is the biological relevance of this, my collaborator has a nice ATPase assay that we used, and he found that there is that GMP, guanosine monophosphate, stimulates the ATPase activity of BIP by around 20%. So it suggests that there is some biological relevance here. Now, BIP is a nucleotide-dependent HSP chaperone. There's a co-purifying nucleotide here, so my first worry was that guanosine monophosphate is going to compete with ATP for access to BIP. That GMP is binding in the ATP binding site. However, that is not the case. So I have BIP that is co-purifying with endogenous guanosine monophosphate, added ATP to this, and there are actually two binding events. There's ATP binding to the apoprotein and ATP binding to the protein that is already bound to guanosine monophosphate. So the KDs for these two binding events are roughly comparable, approximately 2 micromolar in each case, but it's clear guanosine monophosphate is not occupying the ATP binding site. Repeated this experiment with BIP that was purified from BL21 that has no endogenous ligand co-purifying, and the results are roughly comparable, which was reassuring. So BIP has been crystallized before from multiple different species. There's no physical structure for the entire BIP protein from yeast. So I put the sequence of BIP into Chi1, which is it's a contemporary of AlphaFold 3. So I put the protein sequence in there and asked the protein, where is the ATP binding site? The ATP binding site is predicted by the program with high confidence, and the structure of this overlaps very nicely with a previously reported crystal structure, which suggests to me that I'm on the right track here. So looking at adenosine triphosphate, ATP bound in this binding site, ATP is here, and there's a series of residues that form close hydrogen bond contacts with the nucleotide. I went in and replaced all 4 of these residues with alanine, purified the protein. Gratifyingly for me, there is a strong inhibition of ATP binding after making these mutations. So the mutant, whereas the wild type in the presence of 20 micromolar ATP binds the nucleotide very nicely, this mutant, this ATP binding site mutant, barely binds it at all. So that means that this model is right. I subsequently used Chi discovery to search for a binding site for guanosine monophosphate. Unfortunately, there I had less success. So the Chi1 program, it found a number of potential binding sites for guanosine monophosphate, where again, there's the nucleotide bound there, has strong hydrogen bonding interactions with a number of side chains, positively charged side chains. Unfortunately, mutation of these side chains to alanine produced no effect whatsoever on binding. So that was a surprise. That was really one heck of a surprise for me, I can tell you. So, however this is binding, and the AI-driven tools that I've got, Chi Discovery, AlphaFold 3, they're great. But unfortunately, they haven't been able to find this binding site for me yet. So what I'm doing right now is I've gone back to the structure, because honestly speaking, the nucleotide has to be binding somewhere. It's either binding somewhere in this nucleotide binding domain, or it's binding somewhere in this substrate binding. And what's lucky for me is you can effectively cut BIP in half. You can produce the substrate binding domain in isolation. You can produce the nucleotide binding domain in isolation. Try binding assays with those 2 pieces of the protein separately. The expectation being that the guanosine monophosphate will bind to one or the other, but not both. I can then do X-ray crystallography to find out where really this binding site is. And of course, the other question is that because BIP is so highly conserved, is this guanosine monophosphate phenomenon that I observed with the yeast BIP, is that exclusive to yeast BIP, or does it occur in other species as well? So I've produced and purified human BIP. And this protein here is a little more challenging because the full length BIP is shown here in the black charge series centered around the 16 plus charge state. But there's also some truncations happening, such as this, including this major pink one here, shown here with a mass. We're losing almost 2 kilodaltons off full length BIP. And because BIP is truncated, because BIP is partially disordered at the extreme C terminus, it's highly likely that this truncation is the C terminus being removed, being proteolyzed. And because we have such sharp peaks here, this mass is very exact. We can use that mass to map to figure out almost exactly on the sequence where that cleavage site is. So we can produce homogeneous, pure human BIP and do these binding assays as well with the ATP, with the guanosine monophosphate to see, is this novel regulation of BIP by monophosphate nucleotides, is that specific to yeast, or is that conserved across multiple different species? So that brings me to the end of my presentation. I'll hand it over to the other speakers now. Thank you for your attention, and I look forward to the question period later. Thank you.
Thank you very much, John, for an excellent talk. I hope you can hear me, yeah?
Yeah, I can hear you.
Great, great, great. Just confirming. Thank you so much once again. Our second invited speaker is Susanta Chatterjee from Queen's University, Belfast. Susanta you can see him now, is a postdoctoral research fellow exploring the role of RNA binding proteins in anti-cancer drug response. His research career began with postgraduate studies in molecular biology and genetics at University of Calcutta, where he also gained experience as a project fellow investigating the molecular mechanisms of antifungal resistance in crops. Driven by a fascination with the RNA world and its role in human disease, Susanta transitioned the focus to RNA biology. Subsequently, he received a PhD scholarship from the Government of India, allowing him to delve into the study of microRNA-mediated gene repression in immune responses. Today, he will be presenting his work on the ribosome quality control mechanism protects cancer cells against the 5-fluorouracil-induced toxicity. And at this point, I hand over to Susanta.
Thank you very much for the introduction, Kathy. Can you hear me and see my screen?
Yes, I confirm that I can hear you, I can see you, and I can see your slides as well.
Oh, thanks very much. I could just turn on my so I would be sharing my for last few years story about one of the crucial steps of the translation quality control, that is the ribosome quality control. And I try to discover and see how this ribosome quality control impacts in the anti-cancer drug resistance. So to start with, we started working with one of the oldest anti-cancer drug, 5-fluorouracil, which is used for last around 70 years, and which is a pyrimidine analog of uracil, and as I was telling, which has been discovered as a new class of tumor inhibitory compounds in the last 70 years, and that has been still is one of the standard of care treatment for different types of cancer. And 5-fluorouracil is prescribed to millions of cancer patients globally, but the problems comes with its the cancer cells resistance to it. As you could see, the many types of cancer are treated with the 5-fluorouracil mediated treatment as well as combinatorial drug, but the level of mortality rate is so high that it needs a further attention to understand the level of why this drug is showing less efficacy. So we just checked the different mortality rate of different cancer, and the colorectal cancer is one of the highest cancer that is the highest mortality around the globe. And that is one of the reasons is the 5-FU anti-cancer drug resistance. And the majority of the chemotherapy treated for the colorectal cancer patients is based on the 5-FU, as you can see with the data. So we posed the question that how we could increase the efficacy of 5-FU such that the 5-FU treatment can be, anti-cancer treatment can be better. So what do we know about the 5-FU, how it acts? As I was telling, it's a pyrimidine analog, it enters inside the cell, and generally it has been discovered as it inhibits the synthesis of the nucleotides by it inhibited the thymidylate synthase activity. Also, it produces another metabolite, which the 5D UTP, which causes the DNA, disrupts the DNA synthesis and repair, which causes the DNA strand break, cell death, and many other things. And I should comment here that the discovery of 5-FU and all of this has been started as a DNA damaging agent. But later it has been found that the 5-FU can integrate it into the RNA as well, and these RNA damage might lead to the cytotoxicity as well. But most of the research have been focused only on the DNA damaging arm rather than the RNA damaging arm. So we posed the question that what is the mechanism of cellular response to the RNA-dependent 5-FU damage, and mostly the 5-FU resistance and how the mechanism why the 5-FU is showing resistance is mostly focused on the DNA damaging based part. So we were interested to see that does that RNA damaging part has any consequence on building the anti-cancer drug resistant on cancer cells or not. So this has been already previously described by the one of our monumental work by our institute, which I belong, the Patrick G. Johnston's work in the 5-FU. But still, we do not know that 5-FU gets incorporated, what is telling, on different forms of RNA, from ribosomal RNA, tRNA, and many types of messenger RNA, microRNA, and everything. But we do not know that how 5-FU affects the translation and what is the mechanism of these cellular response to these RNA-dependent 5-FU damages. We have started to see that how 5-FU gets incorporated into the RNA. So to do that, we just did the simple immunofluorescence study. We treated the cells with 5-FU, and we could see within 30 minutes, we poked with the antibody specifically for 5-FU, and we could see that the extranuclear localization of the 5-FU all over the cytoplasm. And to confirm the RNA incorporation, we treated the cells with RNAs, and we could see that the level of fluorescence goes significantly down when RNase has been treated. So it shows that the level of incorporation of RNA is very rapid, and it's quite higher than that of DNA. So then we wanted to know that how do cancer cells respond to this damage, and does this contribute to protection or building up of anti-cancer drug resistance? To start with, we first checked the level of protein synthesis inside the cancer cells. As I was telling, mostly the work has been focused on the colorectal cancer model and pancreatic cancer model, because these are the two cancers where the 5-FU has been used as a standard of care treatment. So most of the data will be presenting of the colorectal cancer cell lines. So what we could see that the rapid surge of protein synthesis after 5-FU treatment, although most of the previous publication shows the opposite way, that the 5-FU reduces the global mRNA translation. But most of the data shows in after post 24 hours, 48 hours of after treatment. So there might be many other consequences which hides the RNA-dependent activity because of the confound effect of many other damaging arm inside the cells. We started to see in a very early point, and which shows the time-dependent increased protein synthesis. We wanted to see what happens to the mRNA-associated ribosome. So we did the simple polysome profiling assay. You could see that, I hope you are familiar with that. We isolate the ribosome and run on the sucrose gradient. And then based on the concentration of sucrose, we can get the ribosomal subunits, the monosomes, and the polysomal peak. And this shows the mRNA association with the ribosome, and these polysome peaks suggest mostly that the more association of the mRNA with the polysome suggests more translation is happening. And the protein synthesis data correlates with the mRNA polysome profiling data, as you could see a huge bulk after 8 hours of 5-FU treatment. And also we want to see what happens to the different translation factors. And for that, the mTOR is one of the crucial factors to regulate the translation, as we know. And we could see that the very rapid increased activity of the mTOR, which shows it by downstream upregulation of the phosphorylation of its S6 and 4-ABP1 target. How does the cancer cell respond to this increased translation? So then we just hypothesize that what could be the consequence of this increased translation. So in the normal cells, the mRNA, the normal translation happens, more protein synthesis is going on. But when there is a damaged nucleotide or truncated mRNA, so the ribosome gets stalled. And then the trailing ribosome collide with the stalled ribosome and cause the ribosome collisions. And previously, it has been discovered from the Sunenberg lab and Ramanujan Hegre lab that the ribosome collision is uh, activated by the ZNF598 and E3 ligase, and these causes the ubiquitination of the ribosomal small subunits, and these causes the degradation of many of the protein component of the ribosome as well as repress the translation initiation. So we could question that does 5-FU activates the ribosome quality control? To check it, the ubiquitination of ES10 is one of the markers for it. And then we check how the ubiquitination is changed after 5-FU treatment. And what we could see, the 5-FU treatment rapidly increases the ubiquitination of ES10. So this suggests that the RQC is getting activated. And when we made the RQC deficient cells, this ES10 ubiquitination, we cannot see the evidence. So we could see that the protein synthesis is down and RQC is activated. Then what happens to the RQC deficient cells? Does the translation is again going up regulated here as well? In our polysome profile data, we could see that the polysome is getting upregulated, but when we check the level of protein synthesis, we could not see, like parental, the level of protein synthesis has been reduced in the knockout cells. So is there any ribosome collision after 5-FU induction, and that is being resolved in the parental cells, but in the RQC deficient cells, it cannot be? To check that, we performed a simple ribosome collision assay. So we just treated with the endonuclease. So if we could see there is no collision, then we could not see any peak other than the monosome. But if there is a collided ribosome, like in the knockout, without any treatment, we could see a small peak on the disomes. So then we treated the cells with 5-FU to see that status of ribosome collisions in the cells. And as you could see, in the RQC deficient cells, there is a clear peak of the disome and trisome, which is absent in the 5-FU treated parental cells. And as it has been previously postulated that these RQC, activation of RQC leads to the clearance of the damaged RNA and the proteins, as well as it leads to the inhibition of translation. But when there is a no RQC, it leads to the cell toxicity. So does RQC deficiency has an impact on the 5-FU toxicity? And to check that, we just simply check the level of the drug response after 5-FU treatment. And as we could see in the knockout cell, we could see the high sensitivity of 5-FU. And when we treat it with the specific metabolite, which only incorporated into RNA, FUTP, that also shows a similar observation as the 5-FU. So we could see that 5-FU induces translation, 5-FU increases the collision, which activates the ribosome quality control, and ribosome quality control deficiency increases the 5-FU sensitivity. How 5-FU is causing this collision? Check that. We postulated that this activity is going through the more translation is happening. So that's why we block the translation initiation. And when we blocked with the mTOR inhibitor TORN1, we could not see the ubiquitination of ES10 in the parental cells, just like behaving like a RQC deficient cells. As well as when we treated the cells with the taurine plus 5-FU, we could not see those ribosomal peak other than the monosome, as you could see on the red line after taurine treatment. Those peaks are gone. As well as the sensitivity of the RQC deficient cells goes down, as you could see that the 5-FU sensitivity is quite similar to the parental cells. Then we wanted to see how this is happening inside the cell. Does 5-FU has a direct influence on the RQC factors? And when we see that when we try to check by the expression of 5-FU induced RQC factor, we could see that there's different RQC factors, especially the ZN598 and VY84 shows the upregulation. And when we see the post-transcriptional change, there is mostly no change in the mRNA level. We try to see that what happens to that is that these changes happening at translation level or not. So we inhibited with the cyclohexamide, a translation inhibitor, and these actually, the level of upregulation of RQC could not be showing the difference after 5-FU treatment when CHX has been pre-treated. Then we thought that to directly see that the mTOR regulates the RQC expression, we inhibited the cells with the mTOR inhibitor again, and we could see a dampened expression or upregulation of the ZN518 and BYF to some extent. So you could see that 5-FU induced ZNF518 expression is post-translationally and which is mTOR dependent. So we could see that 5-FU activates the mTOR, induces the translation. This enhanced translation induces the ribosome collision, and that probably leads to the activation of ribosome quality control mechanism. And in turn, 5-FU induces the expression of ZN518, probably, and this is probably the mTOR dependent. And in this way, the activation of ribosome quality control protects the cancer cells from the cytotoxicity. And these works shows how in future the ribosome quality control translation can be also used as a targeting factor for a combinatorial therapy for efficient chemotherapeutic treatment. That's in my future plan work. And as you could see with the, we could scan it and go through the details, and that has been published a few months ago in Nucleic Acid Research. Thank you very much, Mehdi, for all of your support every time. I don't know if you're on. And, thanks very much, as all the lab members, everyone, for the consistent support on that. Thanks all of the collaborators. We are thankful for their support and advices, and I have been supported by Academy of Medical Sciences and other funding. So thank you very much. Thank you, Biochemical Society, for giving me opportunity to present my work. Thank you very much for the invitation. Thanks.
Thank you so much, Susanta, for the great talk. Thank you. You can hear me, yeah? Yeah, okay. Yeah, that's great. And we can now welcome questions for any of the speaker. I can already see that some questions appeared, which is great. If you have a question, please type it in the question box as shown in the image on the screen. Okay, the first question is for John. The question is from Abraham Olavole. And
I know him.
Thanks, John. There is a co-structure of BIP with a ligand bound. How does it compare with the predicted model?
The agreement is very good.
Sorry?
High agreement.
High agreement.
Yes.
Any RMSD value?
I don't have those figures to hand.
Okay. Thank you so much. Okay, then possibly I will ask a question for Susanta. My question would be, you mentioned a little bit your future work, but it was a little bit short. Would you mind elaborating a little bit more and telling us, don't tell us all your secrets, but it was amazing talk. Thank you so much. I just would like to hear a little more on the future directions.
First of all, I tell that just a few days ago, I have been funded with a CRUK grant for just for this follow-up work. So our plan is to understand that how this translation quality control affects the anti-cancer drug efficacy. So I would try to understand different ribosome quality control factors. So here we have been mostly focused on few of them, but there are many others as well. And also there are, as you could see, that how mTOR influences the RQC factor. That is quite an interesting part for us. There are a few observation which tells that the mitochondrial stress activates the Z598 autoextraction that has been published last year, as well as I could remember there are another work on the same nutrient has something to do with the Z598. So we want to understand that how the ribosome quality control is influenced by the anti-cancer drug. Is it what's the upstream molecule, to be very precise on that? So we could try many other, like we try to find out the upstream target if we could, as well as expanding on other ribosome quality control factors, because what I was telling, the 5-FU work or most of the genotoxic drug effect has been mostly centered on the how it causes toxicity to the gene, means the DNA part, not on the mRNA part. So my interest would be how the RNA incorporating phenomena impacts the chemotherapeutic inefficacy. That's the plan.
Thank you very much. The next question is for John, and the question is from Manish Kushwa. For John, excellent work. Can you comment on allosteric effect of GMP concentration gradient on BIP-ATP affinity? This might help you postulate the binding site of GMP to BIP. Apologies if you have shown the data and I missed it.
Yeah, I should actually, in response to your earlier question about the overlay between the crystal structure of BIP and the model generated by AlphaFold 3, the overlap, the similarity is very high, but I should emphasize BIP has been crystallized from numerous different species, numerous times. Never, ever has anyone seen a guanosine monophosphate nucleotide in the crystal structure. So that's the key novel observation in my work, because the ligand is there, but it is quasi-invisible from X-ray crystallography, perhaps by poor resolution. Now, to answer Manisha's question, what was interesting in my assays is I always plot fraction bound observed by native mass spec. Fraction bound is a function of total ligand concentration. And when the enzyme is purified with endogenous guanosine monophosphate, I can see two KD values. There's the KD of ATP binding to ApoBip, and there's the KD of ATP binding to BIP that is also bound to the guanosine monophosphate. And what's interesting is the so-called Bmax, the fraction occupied of when ATP binds to BIP that already is preloaded with guanosine monophosphate. That Bmax value is always higher than Bmax for ATP binding to ApoBip. It's on the verge of being statistically significant, but not quite. So what that implies is there is some modest allosteric communication, a modest allosteric boost, if you like, where GMP boosts binding of ATP. So in the Taiwan models, where I was searching for the binding site of guanosine monophosphate, there were a couple of models where GMP was docked very close to the ATP binding site. There were even hydrogen bonding interactions between the nucleotides. Unfortunately, with site-directed mutagenesis, I couldn't confirm that interaction. So the hunt, effectively, for the nucleotide binding site, that hunt goes on.
I mean, following this discussion, have you ever tried ITC in comparison to your studies?
I have not. It is certainly possible to do because the rate of ATP turnover by BIP is extremely low. So you could even do it by ITC using ATP. You might not even have to do a non-hydrolyzable analog. But like BIP is overwhelmingly expressed in E. coli. It expresses big league, purifies big league. So cost amount of protein is no impediment. That I could try that. Thank you.
Thank you very much. The next question is for Susanta. And the question is coming from Emmanuel Eyafor. Hi, how does your 5FU plus DAPI, I think dye for tracking localization, get into the cell? Is it receptor mediated or simple diffusion? If receptor mediated, which receptor and what mechanisms?
No, it's actually, there is a commercial antibody available for the 5FU. So it has been like a immunofluorescence test. So it's there is a company which produced the antibody for the 5-fluorouracil, and so that has been used as this conjugated with the Alexa flow, and then it has been just imaged in the confocal microscopy and done with collaboration with John, as you could see. I think it's just like a normal immunofluorescence.
Okay, I think we have possibly time for one more question. Okay, the next question is for John, and the question is coming from Alice Baderinva. Thank you, John. Very interesting presentation. A bit of a leap here. Do you think
A bit of a what?
Leap here? Do you think some of this mutation could account for some adverse reactions seen during clinical trial or post-market release, which may not have been identified during preclinical early development? I'm just reading the question.
That's an interesting one. I don't know. So the only reason for making these mutations was to see, does this mutation knock out binding of this guanosine monophosphate? That was the only question. Does the mutation block binding of the nucleotide? Yes or no? That was all I was interested in at the time. I think this question is getting more into like bit function more broadly. Like I showed the diagram showing the mechanism of it. There's a lot of things this protein is involved with. Yeah, in principle, yeah, they might have some effect. Like I talked about the role of BIP misregulation, BIP misexpression in cancer metastasis. So yeah, it's possible that these mutations do something else to BIP, screw something else up. But I don't know. Like there's 685 residues in this protein, and I picked. It was basically an educated guess driven by the Chi-1 modeling. But I mutated residue 165, 213, 238. I don't know. It might. Interesting question. Thank you.
Thank you so much. I think we are more or less at the end of our question time and our Q&A session. On this note, I would like to thank everyone for attending, and thank you so much for the speakers. And you can continue the conversation online. Follow at Biochem Society on Blue Sky and X. We welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. If you have an idea for a webinar in 2025, we invite you to submit a proposal for an upcoming webinar. You can find more information about the webinars at www.biochemistry.org. All our upcoming webinars are listed on our website as well. And if you have missed any of the 1000 plus webinars that have already run as part of the series, or would like to watch them again, please visit our website on YouTube channel. Details will be shown on the screen. The recording from today's webinar will also be available to watch within the next couple of weeks. Finally, I want to highlight that it's more important than ever to stay connected and engage with your fellow molecular bioscientists. Join the Biochemical Society's community of researchers and specialists and take advantage of key benefits, including discounted registration fee for society courses and meetings, exclusive access to wide range of grants and bursaries, personal online access to 2 of their journals, and more. Visit the website to find out more. And I would like to thank you all once again for attending. Thank you once again, our speakers, for amazing and very interesting talks. I wish you all have a very nice day and goodbye.
Thank you very much.
Thank you.
Thank you.